So thank you everyone for attending for our next speaker, who will be Alyssa Rinke, who is presenting the topic 3D bioengineered bone microenvironments to measure tumorigenic properties. Alyssa is a current PhD student in CFM, undertaking a project within SimpleHub, and she is investigating metastatic prostate cancer. So thank you, Alyssa, and when you're ready, please share your screen.
Thank you, Jess. You can see my screen?
Yes.
Perfect. All right. So as Jess rightly stated, my project is looking at 3D bioengineered bone environments to measure tumorigenic properties, specifically looking at prostate cancer. So prostate cancer is the most diagnosed cancer of men in Australia, and it has been for a number of decades. The discovery of PSA, or the prostate-specific antigen, in 1984 has led to a significant increase in incidence because we are able to actually diagnose prostate cancer a lot earlier now. Now, if you look at the comparison to the mortality, that mortality rate is on the decline. And that is because we are able to treat primary prostate cancer early, and generally this causes a very high survival rate. Now, unfortunately, for about 35 to 40% of men, they will actually develop a metastatic prostate cancer, which is a cancer that spreads to another organ in the body. 17% of those metastases, or 17% of prostate cancer patients, will develop a metastasis specifically to the bone. And unfortunately, the 5-year survival rate of a bone metastasis from prostate cancer is only 6%. And that has remained constant over the last decade. So one of the reasons that there is such a low 5-year survival rate after a bone metastasis is because we don't have reliable models to actually investigate metastatic prostate cancer. So the models that generally are used in cancer research and historically have been heavily relied on are 2D models. So what this means is cells grow on the bottom of a flask or on the bottom of a dish. They grow in media, so the red would be a liquid and the cells are growing on the bottom of the plastic. And when you stain them and look at them under a microscope, this is what they look like. So they grow more or less side by side and they stretch out to each other. And this has been instrumental in developing new drugs and diagnostics for prostate cancer. However, it's not working for metastasis research. Now, the other type of model, which everyone is probably familiar with, is animal models. We will discuss that in more depth in a second, but animal models are also not accurately replicating the way that prostate cancer metastasizes in the human. One of the newer methods that is becoming popular is the use of 3D models. So what this is a scaffold is placed into media, or instead of a liquid, this could be a gel, and the cells are grown inside of that gel or around the scaffold. And what you can see is they actually grow very differently. So this is the same type of cell that you see at the top here, but you can see they're growing very differently. So they're growing in tiny balls, or they're called spheroids, and they're kind of clumping together. Now, not only do they very clearly look different, they also have different gene and protein expression as well. Now, it has been identified that cells grown in an environment like this are more similar and have a similar gene and protein expression to cells grown in a human than these 2D models, especially for metastasis research. Now, this is obviously helping to expedite that drug discovery pipeline to give us a better idea of what drugs will actually work when we're treating humans. It is more reliable, but it is incredibly expensive. And there are still some areas that are not being accurately replicated as you would find in a human. And one of those things is actually the strength of bone. So cells will behave differently depending on the strength of their environment. So if you're testing for metastatic prostate cancer to the bone and you're testing that in a gel, the cells are actually going to behave slightly differently because they don't have something as strong to kind of attach onto. So in order to understand why animal models aren't working accurately, we have to have a little bit of an understanding of how bone actually grows in the body. So There are two different stages of bone. We've got the woven bone and lamellar bone. Woven bone is the new bone. It's immature and it doesn't have great structure. So the cells are just kind of dispersed randomly. And you can see in this section here, the dark purple parts, these are the nucleus of cells, and they're just randomly deposited. With our mature bone, there is organization that occurs. So it starts to grow in these concentric circles called an osteon, which looks something like this, and that creates the strength that we have in bone. And again, if we look at a cross-section or a stain, you can see that the cells are actually organizing themselves into these concentric circles, and so this is then our mature bone. So throughout your whole life, everyone currently in the seminar is going through a bone remodeling process. So what this is we've got two different types, or primarily two different types of cells that will allow this to occur. We've got osteoclasts that break down our bone or resorb our bone, and then we've got osteoblasts which form the new bone and form that weak woven bone. Eventually, the bone does mature and become the strong lamellar bone. Now, this is happening throughout our whole lives, and the activity of the osteoclasts, or the cells that break down bone, should be about the same as the activity of the osteoblasts. So you've got no net change in bone density. Unfortunately, in certain conditions, we do have that net change, and metastatic prostate cancer is one of those. So prostate cancer will metastasize to the center of the bone, or it's called trabecular bone or spongy bone. It looks like a sponge. It won't metastasize to this very dense cortical bone on the outside. So when we're talking about porosity, we're talking about the center of the bone here. So when you have got normal activity of the osteoclasts and the osteoblasts and everything's balanced, you'll have no net change in porosity. Your bone will look like this. But if there is too much activity of the osteoclasts, or the cells that break down bone, you might develop osteopenic bone. So the pore sizes get much larger, and that obviously creates weakness. If you have too much activity of the osteoblasts, or the ones that deposit bone, you could get osteodense bone, which means the pores get a lot smaller. And while you might think this would make very strong bone, because it's all that new woven bone, it's not actually very strong. And so the risk of fracture is still very, very high. In metastatic prostate cancer, you actually get a mixed picture. So you'll have some bone that's osteopenic and some bone that's osteodense. So while your overall bone density might actually end up being a little higher, because it is woven bone or that immature bone, you still don't have bone strength, which significantly increases the risk of fracture. Now, putting all of this together, in normal bone homeostasis or normal bone health, you've got equal activity of osteoblasts and osteoclasts, and that is mediated by some of the residing cells. So you've got things called fibroblasts, or cells called fibroblasts, and these help to create the environment of the bone, or the extracellular matrix. Now, in metastatic prostate cancer, the activity of those fibroblasts actually changes. And what we're trying to find out is what causes that change. Why does the presence of prostate cancer cells change the activity of the fibroblasts? And then is that impacting the change in the osteoblasts and the osteoclasts, so the cells that determine how much bone porosity we have? Is that coming from the fibroblast activity that's changed in the presence of prostate cancer cells? So that's ultimately what we're trying to investigate. Now, the reason that we can't do this in animal models, or the reason that animal models aren't working, is because mice don't actually spontaneously produce metastatic prostate cancer like we do in a human. So the earliest model that was used for metastasis research was the Dunning rat model. Unfortunately, it doesn't actually metastasize to the bone at all. So in terms of looking for treatments for this type of cancer, it's not useful. A xenograft mouse model can sometimes be useful. It will sometimes have metastasis to the bone. So this is where prostate cancer cell lines, so these are all different types of prostate cancer cell lines, can get implanted or injected into mice. So sometimes it will have a metastasis to the bone, but it will cause either osteolytic change, so too much bone breakdown, or osteoblastic change, which is too much bone creation, but it doesn't have the mixed picture that you see in humans. And the cells also look different to how they look when they're growing in humans. The other very common model that's used is the TRAMP model. It's another mouse model. However, it will only sometimes cause metastasis to the bone. And when it does, it's only osteolytic. So there's only bone breakdown, there's no bone deposition. And therefore, we're still not meeting that mixed change that we have in humans. So the ideal model that we need is something that metastasizes to the bone, but also has this mixed picture. So we don't want one or the other, we want both, and something that's going to look similar to what we have in humans as well. And obviously, I'm not creating an animal model, but by creating an in vitro model, which does have cells which are morphologically similar to humans, and also by incorporating a number of different cell types, we're able to get closer to that model that we really need to find treatments for metastatic prostate cancer. So to create my scaffolds, I am using waste products, primarily eggshell waste. So eggshell is a agri-waste product, and annually over $1 trillion is spent on the disposal of agri-waste. Eggshell itself, there's over 10 million tons disposed of every year from commercial sites. And that disposal is very expensive. So if we're in Queensland, that is over $100 for every ton, and it also attracts vermin and pests during that process. Now, eggshell has a very similar composition to bone, so it's a valuable resource in this space because I am able to use it to replicate the calcium composition of bone, but I can also convert it to hydroxyapatite, which is part of the bone that gets very hard, and use that as a product to actually make the scaffolds. I am also combining eggshells with chitosan. So chitosan is the compound that is found in crustacean shells. You can also get it from fungi, but you're probably more familiar with the solid shell of crustaceans. And combining that with eggshell will create the strength to replicate bone within the model. So in order to actually create the scaffold itself, I needed to obviously get a lot of eggshells, and I milled those into a fine powder. I found less than 100 micron is working well, and some of that was converted to nanohydroxyapatite. From there, I had to create the ink that can be used in the printer. So I combined eggshell and chitosan with a binder, and then had to test the viscosity of that to make sure it would be usable in the 3D printer. The printer then obviously printed the scaffold, and then further testing is currently being completed. So compressive strength is one of the important ones because we want that to be similar to bone. We also need to look at the hydrophobicity. So when you're doing cell culture, if the scaffold or whatever surface you're culturing onto is too hydrophobic, the cells won't actually adhere. So it can't be too hydrophobic. Completing SEM to look at what the scaffolds look like in terms of porosity and do they represent bone. Cytotoxicity, so if these scaffolds kill the cells, that's obviously not going to be useful for this particular research, as well as stability. So knowing how long the scaffolds can actually sit, and then therefore obviously the shelf life of this product. So testing the viscosity of the bio ink, that was a lot of trial and error, looking at the different ratios between chitosan and the eggshell particles, to something that the printer was strong enough to print, but also something that held its shape long enough to go through the drying process to then become hard. So the ratio that we found is the most useful is 1 is to about 0.5 of chitosan to eggshell protein. And when it is finished being printed, it looks like this. So it's about 1 centimeter by 1 centimeter by about 0.5 centimeter. And in this image here, if you were to touch it, it would be like jelly. So it's still quite soft. Once it goes through the evaporation process, this is what it looks like here. So it does change shape slightly, but it still maintains that porosity. And this is just a different porosity scaffold. So testing a number of different porosities at the moment. When completing SEM of the scaffold, you can see that it is quite a rough surface. Now, this is ideal for cell culture because it gives the cells something to actually grab onto to adhere and start growing. And also you can see the pores were maintained that the printer created. Now, some very recent data, recent as in within the last 24 hours, I have found that freeze drying the scaffolds instead of evaporating the binder results in better porosity. So this is an image of, it is osteoporotic bone, so there should be more bone here than there is. But looking at the general structure, you can see that this looks more similar to my scaffold than the previous one. And when we go in at a higher magnification, you can actually see that the pores are interconnected to one another, which means that cells are going to find it much easier to colonize and move around in the scaffold and start using the calcium from the scaffold to begin growing. So this is something that we are going to be doing a lot more investigation on, but it was a nice finding yesterday morning. Obviously, I've spoken about the strength of the scaffold, so this is something that we are also testing. Human trabecular bone has been reported to have a strength anywhere from 0.1 to 30 MPa. And of the papers which are trying to create an in vitro model replicating the bone microenvironment, they have reported compressive strength of their scaffolds from about 7.75 kPa to 124 MPa. Now, keeping in mind, there are only 4 papers that have actually reported on the compressive strength of their scaffolds. So there are other papers that are doing this and other groups, but only 4 have actually reported on the compressive strength. Now, a lot of the current 3D models that are being used are being printed through plastic. So whether it's PLA or PCL, I have also created some plastic scaffolds just as a comparison, and the compressive strength of those was found to be between 0.3 and 0.35 MPa, which is within that range of human trabecular bone. Obviously, we want our bioink scaffolds to also have a strength within that range. And so that testing and data analysis is currently ongoing. The collaboration with the Toowoomba Hospital has also led us to speaking with the orthopedic surgeons, as that hip bone is actually very similar to our human trabecular bone. And so we are in the process of sourcing and testing that bone for its compressive strength to use as a direct comparison as well. Now, from that point, literature suggested that we would need to actually coat the scaffold, in a membrane or in some kind of extracellular matrix to allow cell adhesion. So keeping in the theme with using eggs and waste products, I actually created an eggshell membrane solution. So essentially that is the eggshell membrane on the inside of an eggshell, and it's very high in a number of different proteins, primarily collagen type 1, but it also has got collagen type 5, elastin, and fibronectin, and all of these are proteins that are commonly used to improve cell adhesion. So the use of this should improve cell morphology, adhesion, proliferation, and differentiation, which are all things that you want when you are doing cell culture research. So after I created that solution, I literally just put the scaffolds into there for about 2 hours, take them out, and then put them into the media with the cells, and then allow the cells to start growing. Now, the initial way that I was creating the eggshell membrane protein was very, very labor intensive. So it involved using forceps to just peel away the membrane from the eggshell. It was very labor intensive. It took about 2 hours for every individual egg, and this is just not viable for industry. So I started trialing different methods. I did try just putting a whole egg into acetic acid, and that did dissolve the eggshell after about 2 to 3 hours, but it's then not using a waste product, and it's not viable for industry because you would need to buy a lot of eggs that would then just be wasted. So the final recipe that I tried was actually just cracking an eggshell and putting the cracked eggshell into acetic acid, dissolving the eggshell itself, and then taking out the membrane. Now, this was very efficient. I could do the whole 18 eggs in about an hour, and it's still utilizing a waste product. And because it is efficient and easy to do, it is also viable for industry. So I obviously wanted to check that there was no difference in cell viability using the 3 different recipes. And from the 24 and 48-hour viability test, so this was using some prostate cancer cells, at 24 hours, there was no significant difference between the viability of the 3 different recipes. And at 48 hours, while not statistically significant, there was a trend for the more viable and efficient method to actually have improved cell viability. So this has given me confidence that I will be able to use recipe 3 and it's not impacting on the cell viability. I also have checked that the eggshell membrane doesn't just wash off when we put it into the media with the cells. So this is an SEM of the coated scaffold. And obviously in the SEM, it needs to be dried. So it started to crack, but you can definitely see that there is a coating there which remained intact. And this is a section, so some very preliminary imaging of the scaffold. So it was sliced into fine sections, and this image is actually from the center of the scaffold. And you can see after 24 hours, there are already cells that had made their way and were able to actually get to the center of the scaffold, which gives an indication that it's not cytotoxic and the pore sizes are large enough that it is able to get to the middle, and then therefore once it grows for longer, we can do more imaging tests to confirm that the cells are proliferating as we would expect. So from this point, obviously at this stage, we haven't replicated that very specific bone environment that you find in the body. So our future testing is to actually combine a number of different cell types together. So HS5 cells, these are the fibroblasts. So they're the ones that create the bone environment, the ones that create the extracellular matrix. And we're going to combine those with prostate cancer cells as well as osteogenic cells. So osteogenic cells are the ones that can turn cells into either osteoclasts or osteoblasts to look at the breakdown or the formation of calcium and therefore bone. And what we're going to do is look at it in 2D versus 3D, so adding in the scaffold, and look at the differences that we observe, given 2D is the current, what a lot of cancer research is completed in. But then we're also going to look at having HS5 cells, so those fibroblasts, as well as the bone cells, without the prostate cancer cells, and compare the two. So what gene changes occur when you add prostate cancer cells compared to when there's just healthy HS5 cells or healthy fibroblasts and healthy bone cells. So what we're going to look at there is gene expression. So see if there's any genes that have a differential or changed expression. And then from there, confirm that there is also a protein change. So with gene expression, when there's a change, that doesn't always translate into a protein change. And a protein change is actually the part that is treatable. It's the part that actually creates change in the body. So we need to also confirm that any change noted through this step is also a protein change as well. Now, you might be wondering, what's the end point for this? So what we can do is actually commercialize it. So once the scaffold has been created and once the model has been finalized, so the scaffold is just referring to the actual bone part itself, and then the model is the addition of all of the other cells, as well as any growth factors. And once that has been completed, then it can be used to test a number of different things. So it could be used in research and development applications. It could be used to expedite the drug discovery pipeline and improve our reliability in terms of will a drug be effective or not. But it could also be used as a teaching or educational product. So universities could purchase the product and use it for their students who are learning cell culture or learning about tissue engineering to look at known gene changes, and then they will be able to assess their ability to actually complete that testing because it's a known change. There is a product on the market. It is focusing on the intestinal cells. So there are a number of different cells in the intestines, but it's a similar concept. So they've created their model, and each one of these, I know it's a very big picture, but each one of these actually fits into a well of a 96 well plate. And you purchase the kit, so it comes with the plate, it comes with the media, it comes with the cells, and obviously instructions. And then you can use that to actually investigate different changes or whatever it is that you're wanting to investigate as an in vitro model for intestinal changes before moving on to animal or human trials. So this is something that is available and something that we're using as a kind of a guide as to where we're heading in the future. And their particular model fits in either a 12 or 96 well plate. So for ours, we're looking at making it fit in a 96 well plate, which is what this one here is. All right, so obviously further materials testing does and is being completed to validate how similar our scaffolds are to bone. We have shown that the scaffolds are biocompatible with the prostate cancer cells, but we need to go forward and do that co-culture, looking at the multiple cell types to look and see if there are any biomarkers of metastatic change, and then also using those osteogenic or those stem cells to look at what's called osteogenic processes, which is the deposition or breakdown of bone. Ultimately, putting all of this together, it will provide greater insight into the development of metastatic prostate cancer, why the body lets that happen, and potentially lead to the development of new therapeutics to help treat metastatic prostate cancer. I would like to obviously acknowledge the university as well as CFM and Simple Hub for allowing me to complete this project. The collaboration with Darling Downs Health and Associate Professor Chris Wall, he is the orthopedic surgeon there, has been instrumental. And obviously, I can't thank my supervisors enough. So Dr. Louisa, Professor Eliza, Associate Professor Polly, and Dr. Jess have been instrumental in allowing me to actually get this far in my project. Thank you. And does anyone have any questions?